So given those introductory comments, I think we should open it up to the audience for comments, questions for the panel, and any other perspectives on this important area as we begin to bring this meeting to its end.
Tanya Zapka from Genentech. So I really appreciate kind of the drug development perspective that was presented by everyone here. And a couple of comments is that I think it's really important that the FDA does provide some written guidance on that. I think that will be very helpful. I think as veterinary pathologists, we understand everything you said, and we try to convince our clinicians of that. But I think without written guidance, sometimes they are still very leery of how the FDA will respond. We also try to let them know that we'd rather have a signal in a dog before a human patient. But again, that's sometimes difficult if they feel they'll be put on a clinical hold. And then I really appreciate everything that was done by Gilead to do some of the primary work. What I would say is in that poll, when the veterinary oncologists want to do some work with a drug that they might get from a pharmaceutical company, if you can identify someone to work with, and that might be a veterinary pathologist or a preclinical toxicologist that understand translational medicine, it'd be good to try and identify someone because they might give you insight into dosing regimens or if you need to hit, IC50, IC90. So your study will be much more convincing to that drug company. So they might re-engage with you. And I've seen study designs, and you do your best with what's published, but we don't publish negative data, right? So, so if you can try and do that as you do your work, I think that's a very helpful way to move forward.
Good morning. I'm Dr. Bernadette Dunham, Director for the Center for Veterinary Medicine at FDA. And I just want to say this has been an outstanding workshop. To have the Institute of Medicine embrace this and bring such excellent speakers as well as the audience participation has been fabulous. The compassion is overwhelming, and we all do appreciate the human-animal bond. Having said all of that, and Chris will follow up in a minute, I just wanted to say that with everything we're gleaning here today, this is something that is so important to us. We do embrace innovation. And yes, we have regulations that we have to adhere to. There's an awful lot of flexibility. And the fact that we can move science forward, and now that we're moving into molecular biology and genomics, that silo between veterinary medicine and human medicine just vanishes. And the more that we can knock that down and work together and share the data, we will have a win-win on both sides. And it's groups like this, it's having IOM bring the best of the best to this kind of meeting that's precedent setting, and I think it's going to continue to move this whole area forward the way I know everybody's wanting us to see this happen. And as we do learn more information that we can share back and forth from not only our pets, but at human side as well, it goes both ways, it's not just one-sided, then we do want to have, at the end of the day, an opportunity to have finally approval, both human and veterinary drug approval, so that then in the hands of physicians and veterinarians, they will have an armamentarium that will really help us now continue to battle cancer. We're living with cancer now. Whether or not we'll always have cures, but we're certainly living with it, and it's courtesy of the development, innovation of where our scientists are taking, understanding the complexity of cancer today. And as we work together, we will get there. So it's really just a big, big accolade from me to the whole group and the whole session. And I want to thank you so much for allowing me to be a part of it. And Chris will talk about some CVM issues specifically on drug approval. Thank you.
Chris Loss, FDA CVM. I just wanted to give a little perspective from the veterinary side and the CVM drug development. And it was touched upon with Gilead. A lot of these preclinical studies, these lab tox studies, are done for these human drugs in dogs. And for any of you familiar with the FDA drug approval for pets, they have to satisfy certain safety and effectiveness requirements. Often, these tox lab studies may be able to satisfy the safety requirements. And in addition, these phase 1-2 studies being conducted in dogs can be used to satisfy some level of effectiveness for drug approval in animals. We have, similar to the orphan drugs on the human side, we have something called minor use in dogs, and drugs can get conditionally approved, like the Paxlovid, the Kineret. They're conditionally approved based on reasonable expectation of effectiveness, with the expectation they work towards full effectiveness over the next 5 years. But so these phase 1-2 studies can be used in these dogs to support for the translational medicine, can be used to support animal drug approval, and we're seeing this as we move forward. So very little extra effort is required from the pharmaceutical companies to support drug approval in animals from what they have from the humans.
Christy Richards from Cornell University. So something we haven't talked about much at all that I'm interested in hearing everybody's, maybe even people not on the panel's opinion on, is immunotherapies, which are becoming a big deal in oncology. And it's my understanding that at least some of those are regulated, can be regulated by the USDA rather than the FDA, including monoclonal antibodies currently being developed by Aratana. And as we move into things like PD-1, PD-L1 inhibitors, and maybe even CAR T cells, how will those be regulated, and what are the issues that arise?
And just as a comment, I think anybody who does immuno-oncology understands that the preclinical models for immuno-oncology are fairly abysmal. They're not very good. And in fact, I think one could look at the companion animal cancers and realize that it is a potentially phenomenal amount of information one could gain from appropriate studies in that species.
Agree.
And I think, as a comment, that one of the points that I tried to make is that we really have a big black box, though, both from a reagent poor standpoint for the dog, as well as a really not well-characterized immune infiltrate in most of the cancers that dogs get. So we have to bring the dog up to speed with the mouse. And until you do that, I don't think those will happen, at least not in a way that's impactful.
We're working on that.
And I just wanted to provide comment from it is a little different on the animal side because we don't have CBER and CEDAR, we have USDA and FDA. And for those immunotherapies like the Aratana, they go through the USDA. And for drugs where there's question, there's a jurisdictional working group between the USDA and FDA to determine, who will regulate those drugs or biologics.
I guess I'll just offer to that, notwithstanding the enthusiasm around PD-1, et cetera, the numbers of patients who are having durable benefit are limited. Understanding who's going to benefit from what and why is unknown. The ability to interrogate that is largely unknown. It's part of what provoked my question yesterday that nobody wanted to respond to in terms of the host tumor microenvironment. But what is it about malignant cells that engenders that sort of local tolerance? How does one break that? What kinds of approaches might one put together to have an impact is profoundly important. And probably the worst possible model to begin to interrogate that is in things with rodents. With, dogs have tails too. I was going to say tails, but these are spontaneous sporadic cancers. And an opportunity to begin to interrogate that in the context of the immune system is not necessarily a part of the tumor. So anything that, I could offer to encourage that pursuit, that would require then bespoke preparation of molecules to give for allogeneic reasons. But there's nothing more bespoke than TCAR therapy. So it is something I would encourage people to explore.
Can I just make one comment
Sure.
on this? Push to talk. A person who shall remain nameless at a company that will remain nameless, but someone who's at a very high level in the research organization at this organization told me that if dogs as a model if dogs, comparative oncology could be a model for the development of immune therapies, then it would be really valuable.
Okay, so this is a question for you, Perry. So coming back to your original or introductory comments, you gave us a list of the perceived and actual concerns that people have voiced about comparative oncology, and understanding that this is going to be one tool in a totality of data that is going to inform decision-making in drug development. It's been my approach or view that the way to sort of weigh against those perceived and actual risks is to sort of quantify or describe the value. So you have some balance around the decision. So I guess the first question, do you agree with that as an approach? And the second is, if that's true, what are the things that you would ask us to do to nail down the value? And Daniel brought up some of the potential sort of low-hanging fruit, but what are the, what's the result that would say this is a good idea?
I don't have a short answer to that. My overall impression, I think you know, Chand, is there is a perception-reality disconnect in terms of the risk. I've never understood moving forward separating risk and benefit. And I just don't know how to do that. And in terms of what I think the biggest gap and the biggest opportunity is, so what does it take to create reason to believe that there is something? So you have this molecule, you're going to administer it, and you want to create some reason to believe. So there's all this work trying to pursue, for example, disease in a dish, to try to recreate that. We exert tremendous effort trying to recreate human disease in rodents. And here we have an opportunity, I would say, where you have sporadic disease, which in many cases is not dissimilar from what occurs in humans. We had big discussions the last 2 days around some similarities in the genetics, and some of the histology is clearly the same. And the heterogeneity is sort of implicit in what we do. And I think the greatest thing that would serve moving forward is that reason to believe that you can give something to an animal or a human that's going to have an impact on the disease process. The fundamentals of, are you hitting the target? Do you know that? And so in hitting that target, you can have an impact on the process. And if you can have an impact on that and a pharmacological effect, and if you can have a pharmacological effect, are you going to have an impact on the disease process? There's no more kind of straightforward way to pursue that than in the context of cancer. And in particular to me, so for most adult malignancies, and now I'm a pediatric oncologist by training, but to me, one of the biggest challenges to address in oncology in general is that of things like metastatic disease. And how do you study that? Can you demonstrate that you're having an impact on those processes alone and in combination? And there's nothing that trumps data. There's nothing that trumps, now this may offend a couple of people in the audience, there's nothing that trumps data that doesn't require a statistician to tell you that you're doing something. So that would be my answer to your question.
Thank you.
Or an answer to your question, probably not a very good one.
Thank you. I have a question for Tanya.
Yeah.
So Dr. Layden presented, in my mind, a very reasonable sort of interpretation of how these data could be or should be or have been viewed by regulatory groups. So I appreciate that. And I guess my question to you is that sufficient in your mind to address the perceptions that exist in your sphere of communities about these potential bad things that are going to happen?
Yeah, so I think we can, on a team-by-team basis, try to educate the teams, but nothing trumps a guideline from regulatory to dissuade. And I understand your perspective because it's going to be case-by-case basis as well, and you're going to look at the totality of the data, and we know that. So I think it's going to have to be a conversation, and I think we talked with Amy LeBlanc about this as well, is that conversation maybe starts between the CVM and the human side of the FDA and the COTC, where the biotech pharmaceutical stays out of that, so it appears, or it is unbiased, so it doesn't often appears that pharma's pushing regulatory to do one thing or another. And I think maybe coming up, not with a hard guideline, but some guidelines would be helpful for us to help educate our clinicians. Often they need external expertise to be convinced, especially when the safety margin between effective dose and toxicity in a dog is quite close, which it can be 1x, which is within PK variability. So these are all discussions that happen case by case as well. But the more support we can put and the more external influence we can offer, the more helpful that gets. But I know that comes with some pain points from the regulatory bodies as well.
It just takes a lot of time.
I know. I know.
Terry Opnorth with CSU Ventures. So in some ways, maybe the immunotherapy comments helped frame what I might suggest, and looking for a response from the panel about. But you know, cancer is cancer in the sense that it's a dysregulation of cell growth, and metastasis, you know, is something associated with most cancers that also has common mechanisms. Yet the comparison between the histopathology of a cancer in the dog versus the human can be quite different. There are not that many examples really where the disease is clinically defined the same way in the human as it is in the dog, which tends to push people back from considering a comparative oncology trial. And so what I guess I'm asking is whether there would be industry interest in something which is more empirical, in the sense of an all-comers trial where you have the opportunity to look in comparative oncology at a number of different cancer types, not necessarily in the same histopathology class as potentially the indication being pursued by the sponsor, but yet one where the mechanisms, the pathways, are probably the same, even if the driver mutation is not. So, you know, can we learn more about the potential of a drug with a little bit more of an open-minded approach to comparative oncology than one which is narrowly and more reductionist in its approach?
Yeah, so that's a great question. Maybe I'll take the first stab at that. So when you're playing in a target that you don't know exactly where it should land, and that it may have wide applicability. So, for example, you know, a tumor-initiating cell or a cancer stem cell target, we tend to think could be applicable in a lot of different tumors. This is a great example of a kind of place where you would take sort of an all-comers companion dog trial. So to do that in humans may be a little bit challenging in the big pharma space because, again, I've reiterated that if we don't show sort of clinical activity in phase 1, you know, we could get knocked out of the ballpark because there's lots of folks behind us trying to take that funding. So you know, more and more in first-in-humans and phase 1 trials, we really try to enrich for the patient population that we think is going to give us that clinical response. But if you're working in, you know, in immunotherapeutic field, or, you know, a great example, I've just joined Fate Therapeutics 2 months ago, where we're working with Juno to augment their CAR T, you know, their CAR T therapies. We've got ex vivo expansion of umbilical cord blood, you know, to use in transplantation for all kinds of diseases, including inherited metabolic diseases. We have an iPSC program. These are all things that, you know, we're looking for a place to land clinically. So that's another great example of, if we can understand the biology of the dog model a little bit more, you know, that could really help augment us to do maybe an all-comers or a select patient population in dogs to lead us to humans. Because, you know, right now, you know, in that field, the human data is lagging. You know, we're really really trying to understand more fully where to place our bets.
Yeah, just to be clear, I'm not suggesting that you would change that strategy. I'm really suggesting a parallel strategy where you would use the oncology all-comers to take a long view for your cancer development program. You might be literally focused on that target and selecting a target-specific phase 1 program to launch into, but looking wider using the comparative oncology approach that might inform your next development decisions in terms of what other indications you might pursue.
Exactly. Again, sort of I pointed out, it can be an iterative process where you go back and forth. So,
yeah, I think there are a variety of modalities we use, pharmaceutical industry uses, to evaluate across a broad panel of tumor types. There are CROs that can run panels of 200, 300, 400 cells, not just as monotherapy, but as combinations. I think one can't approach the companion animal model, companion animal cancers, without a hypothesis. So I would not suggest that anyone throw a blind net as an all-comers of looking for things. I don't know if that's what you're proposing or not, but I think we really have to understand what we're doing, and there has to be a very well-defined hypothesis of not just an all-comer, but I would take the opposite approach of what defined subset of canine patients have the pathway that we think our molecule will target. This is how we got to targeted therapeutics. It wasn't by luck and happenstance. It was by dissecting the tumor pathways and understanding the resistance mechanisms and genomics. And if we apply that same methodology to canine patients, we'll be able to interpret the data and also interrogate that system in a way that will be successful, I think.
There's something to throw out to this audience. There's something called an N of 1 trial design. So in aggregate, those data can be informative. And just as maybe a provocative proposal, I wondered, as a way to kind of address heterogeneity and the ethical considerations and the cost attendant to doing studies in companion animals, maybe like pediatric oncology, where the standard of care becomes participation in a clinical trial and the accumulation and the collection of clinically relevant data, which can be analyzed and sometimes analyzed in aggregate across, may be an interesting path forward for this community. I don't know, but this N of 1 kind of design is something I would have a lot of time for. So if there is a particular pathway, for example, activated, PI3 kinase pathway activated, evidence for that in a particular cancer, a particular animal, and you interrogate that, you have a pharmacological effect, and you have something that could lead you to then other studies where you see this pathway activated. And I think that is something to really consider, but that obligates then those that pursue that to collect those data, to share those data, and to then pursue additional experiments subsequent to that, both in other pets and in humans.
And those
may be an interesting idea.
Those are being pursued now as basket trials. And so there's several pharmaceutical companies that are not going after the cancer type, but going after the target biology, and they're trying to understand if that will work. And that gets to looking across disease types or cancer types based on target biology, but that means you have to understand the target biology.
Yeah, Carolyn Henry, University of Missouri. I'd like to start out with a question to the crowd instead of the panel, if that's okay, and just ask, by show of hands, how many of you have heard of the concept of one health, one medicine? Okay, that's your blue sky right there, because I asked this question last year in this city with a similar crowd, and there were fewer hands going up by far. So I do think we're making some progress. I also think we've busted a lot of myths here in just a day and a half. I wrote some down as they happened. First of all, John, I appreciate you trying to bust the myth that we're going to see a lot of adverse events in animals that will shut down trials, because it's my perception that that's relatively uncommon as well. There was not quite the myth, but the thought that we don't publish negative data. In veterinary medicine, we may not publish it, but sometimes we celebrate it, and we always try to figure it out. And I think Cheryl gave us a really good example yesterday of an instance where an extravasation of a drug resulted in something that was very telling for moving that drug forward. So, I think we all want to have the outcome that we expect when we go into the research, but in reality, most of the really big things that have ever happened in the field of medicine have happened by looking at mistakes and trying to figure out why they happened. As far as standard of care, that was brought up a lot in reference to veterinary patients. And just for those that aren't in the veterinary field, what you need to understand is we have less than a handful of approved cancer drugs for use in animals. So when we say standard of care, we're not saying approved drugs. We're saying human drugs that we're using in animals the way that it looks like they work the best. So I do think we have more flexibility, and I do think that's something that we can use to our advantage to try to create new trials. And then finally, I just wanted to finish up with saying that I applaud Rod and everybody that worked to bring this together. I'm in an area where I work in One Health, I work in oncology, I work in the black space called administration a little bit. And there's nothing I love more than seeing groups like this come together, and I would challenge each and every one of us to at least follow up with 2 people that you met from this meeting. Don't just take their card and go home and wait till the next meeting. We got to keep this conversation going. If you need people to talk at conferences, I can tell you that all the oncologists on the veterinary side will likely say yes to any invitation that you give us. For those of you that want to write articles and help spread the word about this, we would love to do that. And if I would ever show up at a meeting and not at some point get the question, oh, do dogs get cancer? I would consider that a really good accomplishment in my career. And then finally, from an academic standpoint, we've talked a lot about what we can do with industry. I think in our setting, we need to streamline the process and make sure that it's not so cumbersome that industry doesn't want to work with us. And from the industry side, if you could help us demonstrate that you get a positive return on investment when you work with animal models, that would be very compelling and something I think should make its way into the literature. Because I think if you did a cost analysis, return on investment, you're either saving a lot of money by not taking drugs forward, or you're making more money by moving them along the pipeline more quickly. So while we look for examples of mistakes, I'd say let's look for examples of where we really demonstrated a return on investment. So that wasn't really a question other than, can you tell me what the return on investment is?
I'm Susan Braun from the V Foundation for Cancer Research, and I have loved being here and being a part of this meeting. I came here originally with my funder hat on because this is an area that we're having some growing interest in for a lot of reasons. But I find myself reacting a lot with my cancer patient advocacy hat on, because I've done that for 25 years. And one of the things that I would just, this is not a question, it's a comment, encourage people is to learn from cancer patient advocates who have done this, our warriors who have done this for 25, 30 years. Learn some of the lessons about IRBs and about clinical trials and about regulations and all of those things, because I think you will find that there are many who have learned over the years that you can easily put too many rules in place and too many regulations in place and things that are going to slow you down. Patient advocates who are on clinical trial review panels and design of clinical trials, I think are there to help expedite and move things forward. Not the way it was at first thought, oh, these patient advocates are going to want to hold us back. Au contraire. So I would really suggest adding people in who have walked this road for a very long time with some very similar issues, and I think might be able to help us move the field forward pretty quickly.
Jaime Modiano from University of Minnesota. So thank you for a great session and for a great meeting and for inviting us. A quick comment for Perry. I think that your own comment about attacking the tumor niche is really important, and especially as we move forward with biologics and immunotherapy. I think the concept of treating this like firefighters, you have to drop some water on the fire, but you also have to create a fire break. And if you make the niche inhospitable, that's an area that might be very successful because the tumor can't mutate to grow more trees that quickly. But a question for the panel, perhaps Anne, Wendy, and Daniel can take this. We heard a lot about this pathologic fear of adverse events, and I think Tanya responded really well, which is you can say anything you want, but until we see it in writing. But there's another part that hasn't really been addressed, and that is when you're thinking, and maybe Daniel can talk about this because they tried it. When you talk about parallel development, how do you ascertain the risks? How do you evaluate the risks of being able to put something that could go into veterinary medicine and into human patients, perhaps using the same molecule or the same compound if you might not have two versions, in terms of being able to then assure that you're going to be able to get reimbursed and third-party payments to agree that treating a people patient costs $80,000, whereas treating a dog that's being paid out of pocket might have to cost, $6,000 or something like that. So, I mean, how are you guys approaching that? Because I think that is another huge hurdle in terms of this parallel development, right? We need to make sure we don't break the business model and that we respect the big market.
So you had a couple of questions. Maybe I'll hit the latter first. So Gilead Sciences does not develop animal drugs. We're not in the business of doing that, and nor was the intent of this particular molecule for veterinary use. so we don't have that dual paradigm. I think the other comment you made was related to the safety assessment during these particular types of trials. I think they can be very impactful for human clinical development because, in fact, they are in disease patients. And as most people know, disease patients, whether they're a canine or a human, actually respond to drugs much differently than either normal healthy volunteers or laboratory animals that are actually very healthy. Typically also for oncology drug development, the requirement for the duration of toxicology assessment is quite short for most cancer drugs. Yet in this setting where one can do multiple cycles, follow dogs, post-treatment, et cetera, you can gain a lot of information on what are the potential chronic safety issues that might arise with this type of drug development. And David alluded to one of them that we actually identified with this type of a class of molecule, which is very impactful and told us what we should be monitoring for in patients. So I think you have to take this not as what's the big bad wolf out there that you might jump into, but actually you want to go find that big bad wolf. And the sooner you find it, the better. We're in the business of developing drugs for patients and transforming their lives in a good way, not a bad way. And if we can identify the pitfalls of our therapies up front, that's of benefit. And I think the agency has embraced that exact approach because, in fact, every finding we identified in our studies with this particular molecule, not just the good ones, but all the bad ones, were recorded very accurately and given to the FDA as part of the IND on a regular basis. As would any new finding in a toxicology study be reported, we did similarly with any new finding we did in these patients with dogs. And in fact, the agency accepted those findings as non-controlled study, very informative, impactful for what we should be looking for in patients, but there's certainly nothing beyond that. So I think it was a really win-win scenario.
Oh, sorry. The question of making drugs available for dogs, unfortunately, I have to say that is a very hot potato, and our approach has been to try to stay away from it because I think it could create a real obstacle for pharma companies, and we need to prove that comparative oncology is a very valuable tool for developing drugs first, and hopefully that problem will solve itself. I think that there are a lot of companies moving into the space of trying to develop drugs for animals, so I hope that eventually they will provide a solution.
Access and reimbursement is a complex issue. I don't know if the analogy is relevant, but making a medicine available in the United States with the access reimbursement processes available is not the same for somebody in India, say, and global multinational companies find ways to manage that. And I wonder if there may not be some opportunities here to explore the implications of that. I don't know, but somebody did mention here earlier the impact of insurance and et cetera. I wonder if some of that might not need to be explored. I don't know, but it is a complex issue, obviously.
Maybe, so a great example of this is, Pfizer bringing forward Sutent and Palladia. Cheryl had a huge role in that. And I think that, hopefully as you bring a lead molecule forward into development, you have several candidate molecules. And, oftentimes it's really hard to pick which one you're going to bring forward into humans. And as it turns out, they may have just a hydrogen difference or something very similar, but the activity is the same, and you could pick anyone forward. And, the case of Pfizer back in the day, we were fortunate because we had a large animal health, group as well that was led business development-wise by a woman by the name of Dr. Haven, Michelle Haven, that some of you may know. And, so we as clinicians were able to partner with the animal health group. So, that was a win-win for everybody. But that was a unique situation in terms of, the company, pieces that it had at the time.
Carol Robertson-Plew with Eli Lilly, and I'd like to just comment on this topic as well. I guess I see great things going forward for One Medicine, for potential new medicines in oncology for animals as well as for humans, and I think it's going to happen. We're watching a lot of small companies pop up across the world right now, working on some novel things in oncology. And I'm on the big pharma side, but I'm watching these companies, and at least one of these companies is going to succeed, and they're going to change the paradigm. The other thing that we need to keep in mind is that drug development in animals doesn't have the same cost associated with it as human drug development. So although the income is fractionally lower, the cost of drug development is also fractionally lower. So all of those things need to be factored in. And I think in the future, it's not going to happen tomorrow, but in the future, we're going to start to see some other things happen in veterinary oncology and things that are available in the marketplace for animals. That's my futuristic look at the world. So I think this whole comparative space is going to open up opportunities for new things to happen. Not every single molecule that succeeds in animals will be taken into humans for a variety of reasons. Maybe we see something untoward in humans that we didn't see in dogs. Or maybe there's a separate molecule, like Wendy just said, that could go in that direction. So those are my thoughts. I'm an optimist.
I just want to follow up a conversation I had with Mike Casson yesterday in the hallway. And it's just to get it on the record. And that is a burgeoning frontier kind of in oncology right now is metabolism. It's not been mentioned at all really at this meeting. And I think the spontaneous animal tumors are just an outstanding model, if you want to call them that, for looking at tumor metabolism and drugs that might interfere with that. So I just want to make sure that's on the record and that people are thinking about it, because I think it's really a win-win opportunity.
Masa Tamura, Kansas State. I came here, I'm a basic cancer biologist. I came here and in the first session, this one medicine is to fill up the gap, accelerate clinical trial, and one medicine has a great hope or bright future. But in the session 2, when I listened to session 2, the landscape of genomics of canine disease, canine cancers are quite different. It's very breeder specific, and they don't share many things with human cancer. So, but apparently overwhelming people here having great hope. So I want to bring back what am I supposed to say to my colleagues in the cancers. So is one medicine still have a great future, or we have to reconsider, is this very species disease specific? If anyone, not only panel speakers in yesterday's or even anyone, can give me some sort of input, I would really appreciate.
To clarify the question, is whether can you just kind of clarify the question? I'm sorry.
Landscape of the genomics of canine disease, canine cancers are quite different from human cancer. But nobody talk about that.
Can I actually answer the question? So it's a very similar on the genomic level. So the way, I guess Dr. Breen is not here anymore, but the thing that what he shows is that, because the cancer genome contain many changes. Some of them are drivers, some of them are passengers. So the way that what his data showing that the drivers are shared between the dog and the human. The dog actually can be used to identify drivers among many genomic
Some
changes.
example, some type of cancer drivers are completely different. Also, the cancer latency is quite different. microenvironment is different.
Well, I mean, there are other things that I guess genome just one level, but there's like an epigenome, microenvironment, metabolism. I think if you're looking for the pathway level, actually dog and human are very similar. I mean, many pathways are shared. So we're showing that in the breast, in the colon, in the head, like squamous cell carcinomas. So actually the molecular level, in terms of the cancer-driven pathways, I think they are shared very much. I mean, as long as the histopathological level, histopathology are shared, I think the molecular drivers actually are shared between the two species.
I would agree with that as well, is that I think that if we take histology off the table sometimes, we have, I think the BRAF mutation is a great example of where there's an identical mutation found in a large number of dogs with a specific cancer that isn't the same cancer, but it's probably likely that the mechanism of involvement of that mutant protein in the biology of TCC is similar to what occurs in colon and thyroid. And the KIT mutations that occur in dog mast cell tumors with a relatively high frequency are very similar to those that are found in GIST. So I think if we take histology out of the picture a little bit, we can find the same pathways, the same mutated proteins, the same tumor suppressor proteins that are driving cancer. And so I would disagree with the assessment that the genomics aren't similar. We certainly have lots of evidence that microenvironment is similar. I think Steve Dow would agree that much of the work that he's done showing macrophage monocyte infiltration into tumors and the various suppressor MDSC, Treg populations that are there. So I guess I think many of us who've done research in this field would disagree with your assessment.
Thank you very much.
I'm mindful of the time. Maybe one final question. You've been very patient.
Well, I just want to second what was just said about similarities between the disease processes in dogs and humans. And I think one of the biggest roadblocks for moving forward to try to find what the underlying disease processes are, for at least for what I'm doing, is having the appropriate canine-specific or cross-reactive molecules. And so if there's any way we can work together to develop those tools that are very important, I think that would be great. And then I just have one last comment, and it doesn't apply to this session, but the last one, and that is that one of the tools that I use when I am talking to owners and trying to explain about the clinical trials and what's involved is that oftentimes, what I'm saying is my experience, but to owners it sounds very optimistic. And so they often ask if they can talk to an owner whose dog has experienced what their dog is going to be going through. And I never have a problem with any of the people whose dogs I've treated in the past. They're more than willing to speak to people whose dogs will be undergoing the similar therapies.
Thank you for your attention to this session. I think we need to move into the final roundup of the 2 days. Thank you. Thanks to the speakers.